Welcome all for the recall and revise series. This series is going to be very important from the examination point of view. And today we will start with the biochemistry recalls. Okay, Dr. Sansi, can you read this first question?
Yes, sir. The parenchymal cells of the liver can convert fructose to glucose, synthesizes urea, conjugates the bilirubin, excretes bromosulfothioline, BSP, synthesizes cholesterol.
True or
Okay.
false type of questions. All are true or false type of questions, and these are the options which are given.
Hmm. It synthesizes the cholesterol. Okay. Convert fructose to glucose. Yes.
Look for
Urea.
100% true, look for 100% false, and then correlate.
Hmm.
And we are talking about parenchymal cells of the liver. Liver has got multiple functions,
Hmm.
and main is the synthesis.
It doesn't conjugate bilirubin. Sir, option 4 and 5 is correct. It also does convert fructose to glucose. It also synthesizes urea, but it doesn't conjugate bilirubin. It is conjugated in
Where does the bilirubin
option,
conjugated?
sir, D.
It is option A. All are correct. All listed functions are performed by hepatocytes. Fructose to glucose via hepatic metabolism and gluconeogenesis pathway. Okay. Then this gluconeogenesis is from non gluconeogenesis pathway is there that you should always keep ready on, and TCA cycle also you should be ready with. Urea
Hmm.
synthesis occurs exclusively in hepatocytes via urea cycle. Okay. Bilirubin
Hmm.
conjugation via UDP glucuronidase transferase in hepatocytes. BSP secretion, hepatocytes uptake and excrete BSP into bile. Liver function test and cholesterol synthesis, major site of HMG CoA reductase pathway. Okay, so all are true.
The
Next,
conversion of glucose to lactic acid, okay, occurs in a single enzymatic reaction. Is the only pathway for synthesis of ATP in the RBC. Is reversible process in skeletal muscle. Is a reversible process in a skeletal muscle. Is inhibited by a high cellular concentration of the ATP. Is inhibited by high cellular concentration of the ATP. Okay, occurs in skeletal muscle when the availability of oxygen is limited. Occurs in the skeletal muscle. Yes, true. Fifth one is true. Fifth one is true. Fourth one is also true. It's a reversible process, and no, it is not true. Third one is false, sir, and first one is also false. So, is the only synthesis in the RBCs. Sir, option B.
option B occurs in a single enzymatic reaction. It is a false sentence. Glycolysis is a multi-step pathway. Only the final step, pyruvate to lactate via LDH, is a single enzyme reaction, not the entire process. Okay, is the only pathway for ATP synthesis in RBC. That is true. Okay, RBCs lack mitochondria, so ATP is produced exclusively by anaerobic glycolysis. Okay, is a reversible process in skeletal muscle. This is false. The overall conversion of glucose to lactate is not a reversible in vivo. Only pyruvate to lactate step, LDH reaction, is reversible, not the complete pathway.
Hmm.
Then is inhibited by a high cellular concentration of ATP. That is true. High ATP inhibits phosphoroctokinase 1, okay, reducing glycolytic flux, and occurs in skeletal muscle when oxygen is limited. Yes. Under hypoxic conditions, skeletal muscle relies on anaerobic glycolysis, leading to lactate formation. Next.
Uric acid is formed from the breakdown of purines. Is raised in serum during normal pregnancy. Is raised in serum during thiazide diuretic therapy. Is reabsorbed in the proximal renal tubule. Is excreted unchanged in the urine. We all are true. So it is a
So
breakdown.
option
Option A. Option
here
B.
they have given, what is B? Is raised in
Raised
serum during normal pregnancy.
is excreted more.
So purine catabolism oxidation results in the formation of uric acid via xanthine and hypoxanthine. The reaction is catalyzed by xanthine oxidase. Pyrimidines are broken down in the liver by reduction to form amino acids beta alanine and beta amino isobutyrate. Uric acid is the final product of purine breakdown in humans. Uric acid is 100% reabsorbed, then secreted in PCT and is excreted chemically unchanged in the urine. Pregnancy levels, they have not mentioned anything. What happens to this serum? Okay, check it. Okay,
Hmm.
so A or B. Okay, next.
Hmm. Adenine is a pyrimidine base, forms base pair with thymine in RNA, is synthesized attached to ribose phosphate, can be converted directly to a nucleotide by the action of phosphoribosal transferase enzyme, is regulated by a pathway which involves the enzyme xanthine oxidase. Sir, option B.
Option B. That means you mean to say 1 and 2 are
Wrong.
false. Option A, E,
E.
E. Okay. C is also false. Third sentence is synthesized attached to ribose phosphate. So there is little discrepancy I have noted. That is why I was also not sure about this particular sentence. So that is why I marked. So I might have confused between B and E only. Okay. So adenine is a purine base that forms a double bond with thymine. So it's a purine. Okay. Adenine is a purine base. It forms a double bond with the thymine in DNA and double bond with uracil in RNA. Okay.
Hmm.
Adenine forms adenosine or nucleoside when attached to ribose and deoxyadenosine when attached to deoxyribose. It forms adenosine triphosphate nucleotide when three phosphate groups are added to adenosine. Adenine is the base, that is adenine plus ribose is adenosine. Adenine plus ribose phosphate is AMP. De novo AMP production involves the enzyme phosphoribosyl transferase APRT. Okay, and production from adenosine involves 11 steps. However, the salvage pathways involve the formation of AMP through the transfer of adenine to PRPP, that is 5-phosphoribosyl alpha pyrophosphate, mediated by APRT. With the release of PPI, it is degraded to uric acid. Okay, next.
Ethanol consumption in excess stimulates fatty acid oxidation, suppresses arginine vasopressin secretion, promotes gluconeogenesis, is oxidized to acetaldehyde, is metabolized predominantly in the liver. Hmm.
Look for three correct, two correct, look for negative, false. Which are you are sure about? Ethanol.
Sir, option B.
Option B. Consumed in excess stimulates fatty acid oxidation is false. Ethanol metabolism increases NADH NAD plus ratio, which inhibits the fatty acid oxidation and promotes fatty liver. Okay. Then suppress arginine vasopressin secretion. That is true. Ethanol inhibits hypothalamic release of ADH, leading to diuresis. Promotes gluconeogenesis is false. High NADH shifts metabolism toward lactate formation and inhibits gluconeogenesis. Risk of hypoglycemia and lactic acidosis. Okay. Then it is oxidized to acetaldehyde. That is true. Ethanol is the first metabolized by alcohol dehydrogenase to acetaldehyde. Is metabolized predominantly in the liver. That is also true. Major metabolism occurs in the liver via ADH, CYP2E1 and catalase enzymes. Next. Yes.
Concerning prostaglandin, arachidonic acid is the precursor of prostaglandin biosynthesis. PG synthase cyclooxygenase catalyzes arachidonic acid to prostaglandin endoperoxides. NSAIDs drug inhibits prostaglandin dehydrogenase. Mefenamic acid is a more potent inhibitor of prostaglandin synthesis than aspirin. PGF2 alpha is excreted unchanged in the urine. Hyaluronic acid option is correct. Okay, option B is correct. And says inhibits prostaglandin synthesis. Option 3 is wrong, sir. Mefenamic is more potent now. So, sir, option B.
It is B. Arachidonic acid is the precursor for prostaglandin biosynthesis. That is true. Prostaglandins are derived from arachidonic acid, 20 carbon polyunsaturated fatty acid released from membrane phospholipids. Then PG synthase, cyclooxygenase, catalyzes conversion to PG endoperoxidase. That is also true. COX-1 that is and COX-2 converts arachidonic acid into PGG2 and PGH2 endoperoxides, and NSAIDs inhibit PG dehydrogenase. That is false. NSAID inhibits cyclooxygenase. Okay, not the PG dehydrogenase, not prostaglandin dehydrogenase. Mefenamic acid is more potent inhibitor than aspirin. That is also false. Aspirin is a strong irreversible COX inhibitor. Okay, so mefenic acid is comparatively less potent and reversible. PG F2 alpha is excreted unchanged in the urine. That is false. Prostaglandins are rapidly metabolized in the lungs and tissues. They are not excreted unchanged. Okay. Next.
Triglycerides contains glycerol combined with three identical fatty acids, may accumulate in liver cells, are present in intestinal cells, are hydrolyzed by pancreatic lipase, are not present in chylomicrons. Fifth one is false. One is obviously false. First one is correct. May accumulate in liver cells. Third one is here. Option E. No. May accumulate in liver cells. Just a second. Sir, option D.
Option
D.
E.
E D D.
B. It is B is correct. Okay. So contain glycerol combined with three identical fatty acids. That is false. TGs typically contain three fatty acids which are usually different, not identical. Okay. May accumulate in the liver. That is true. Excess TGs accumulate in the hepatocytes causing fatty liver. That is hepatic steatosis, and are present in the intestinal cells. That is also true. Enterocytes re-synthesize TGs after absorption of fatty acids and monoglycerides. This all we have seen. Are hydrolyzed by pancreatic lipase. That is also true. Pancreatic lipase breaks TGs into monoglycerides and free fatty acids in the small intestine. Are not present in the chylomicrons. That is false. Chylomicrons are rich in TGs and which form their major core lipid component. Next.
RNA contains deoxyribose, is composed of two nucleotide units, is the main constituent of human chromosomes, is the main constituent of ribosomes, is required during protein synthesis. 4 and 5th is correct. Okay. So, second is also wrong. Third is wrong. Main constituent of fourth and fifth is correct. Third is wrong. Second is also wrong. Option B, sir.
Yes, B is correct. So contains deoxyribose, that is false. RNA contains ribose sugar. Okay, whereas DNA contains deoxyribose. Okay, is composed of two nucleotide units. False. RNA is a single stranded polymer of many nucleotides, not two units. Okay. Then is the main constituent of human chromosomes. That is false. Chromosomes are primarily composed of DNA and associated histones, proteins, and not RNA. Is the main constituent of ribosomes. True. Ribosomes are made up of ribosomal RNA and proteins, with RNA being the major structural and functional component. Is required during protein synthesis. True. RNA is essential in translation. mRNA carries genetic code. tRNA brings amino acids. RNA forms ribosomal structure and catalyzes peptide bond formation.
In 1 turn of a TCA cycle, tricarboxylic acid per acetyl CoA, 3 molecules of carbon dioxide are produced per acetyl in
One ton of TCA cycle.
1 ton of TCA cycle per acetyl CoA. Okay, one acetyl CoA. Then 3 molecules of carbon dioxide are produced. Reduced NADH are produced. Reduced NAD, reduced NAD. That is NADH are produced. Yes. Reduced FADH2 are produced too. GTP are produced too. Acetyl coenzyme is used. Okay, yes. Acetyl CoA are used. CO2 is produced. Yes. Acetyl coenzyme A is also used. So all options are correct actually. Option A.
Option B. And I don't know.
NADH produced.
Three molecules of CO2, 3 molecules of NADH, 3 molecules of
Okay.
FADH2. Okay,
Mhm.
3 molecules of all are not produced, no?
Okay, okay. We got it, sir.
So which 3 molecules are produced?
Only NADH3, one
NADH,
FADH2,
yeah.
one GTP.
Okay.
2 molecules of this.
So just be careful while answering. So per 1 ton of TCA cycle or per one acetyl CoA, CO2 are produced. False. Only 2 CO2 molecules are produced per ton, not 3. Okay. NADH are produced. Yes. Three per each ton produces 3 NADH molecules. FADH2 are produced. False. Only one FADH2 is produced. Okay. GTP are produced. Only one GTP is generated. Acetyl CoA are used. One acetyl CoA enters the cycle per ton, not 3. Okay.
Hmm. Yes, sir.
Nice. That way
Lactose
the
is
question was not difficult. The careful reading was required. Okay. And you should know all the outcomes. Remember, the all cycles and these, they are not expecting in detail, but they expect you overall outcome and the rate limiting steps and all. Okay. Important,
yes.
important. Next.
Lactose is a non-reducing sugar may be detected in the urine of a lactose is a non-reducing sugar. Okay, it may be detected in the urine of a pregnant woman. Is a major constituent of a seminal fluid. Is a galactosyl glucose is catabolized by the liver. Is a non-reducing sugar. First one is false. The second one is also false. Hmm. Lactose may be detected in the urine of a pregnant woman. And
Seminal fluid. What is the major constituent of
fructose.
which? So this is going to be true or false.
False.
So how
Okay.
many options are remaining?
So
B, C, D.
third BCD, yes, sir.
Then now,
Is it is catabolized by the liver or not? It is
is
not.
it a non-reducing sugar?
It is a non-reducing. No, sir. It is a reducing sugar, no.
So B and C will remain then.
Ah.
Then,
Then
then what is the difference between B and C? Only one. So
one. No, galactosyl glucose. Yes, yes. Option 4 is correct. Option B. Galactose.
So is a non-reducing sugar, that is false. Lactose is reducing disaccharide because it has got a free anomeric carbon on the glucose moiety. Then may be detected in the urine of pregnant women. That is true. Lactose can appear in the urine during pregnancy due to increased synthesis and physiological lactoseuria, especially in late pregnancy and lactation. Is a major constituent of seminal fluid. That is false. The main sugar in the seminal fluid is fructose, not the lactose. Is a galactosyl glucose. That is true. Lactose consists of galactose plus glucose, beta 1 4 linkage. Hence, galactosyl glucose disaccharide. It can. is catalyzed by the liver? False. Lactose is primarily digested in the intestine by lactase into glucose and galactose, not metabolized as intact lactose in the liver. Okay. Next.
Concerning carbohydrates, sucrose is disaccharide of glucose and fructose. Cereal grains contains less than 40% starch. Cereal grains. Cellulose is a fructose polysaccharide. A normal diet contains less than 60 grams of carbohydrate daily. A dietary carbohydrate is oxidized in the body to carbon dioxide and water. Is oxidized in the body to carbon dioxide and water. Sir, option A is correct. Fourth is also correct. Fifth is carbohydrate. Actually, for ATP it also generates. So
Which correct? First is correct and
first is correct. Fourth is also correct, sir.
so option, look at the option then. First is correct, fourth is correct. So A, then directly
E.
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 Okay.
Hmm.
A and E.
Hmm. Fifth
Then
is also correct. Actually. Cellulose.
if the
Sir,
fifth is
option
correct,
A.
option B. Okay. So what
And
is the
fourth is
sucrose
actually
is a disaccharide? Yes, it is a disaccharide glucose and fructose. Okay. Cereal grains
fifth.
contains less than 40% of starch. Okay, sucrose is a disaccharide of glucose and fructose. Sucrose composed of glucose plus fructose linked via glycosidic bond. Cereal grains contain less than 40% starch. False. Cereal grains, rice, wheat, maize are rich in starch, usually 60 to 70%, not less than 40%. Okay. Cellulose is a fructose polysaccharide. That is false. Cellulose is a polymer of glucose beta 1,4 linkage, not fructose. Okay. Then a normal diet contains less than 60 gram of carbohydrate. That is false. Normal diet contains approximately 200 to 300 grams of carbohydrate per day, depending on intake. And dietary carbohydrate is oxidized in the body to carbon dioxide and water. That is true. Complete oxidation of glucose yields CO2 plus H2O plus energy. Next.
Energy. Muscle glycogen metabolism cannot yield through free glucose. Metabolism is independent of the enzyme phosphorylase. Metabolism only generates ATP under aerobic condition. Is entirely intracellular. Is released into the circulation in response to glucocorticoids. Glycogen is released into the circulation in response to glucocorticoids. It actually causes glycolysis. Metabolism generates ATP under aerobic condition. No, though it can generate both. Third is wrong, sir. It can generate ATP under both aerobic and anaerobic condition. Cannot yield free glucose is true. Muscle cannot undergo gluconeogenesis. So one is true. So A, C, D. And then metabolism only generates ATP under aerobic condition. It's false. Metabolism is independent of phosphorylase is required, no enzyme phosphorylase. Intracellular released into the cell response to glucocorticoids. The option C.
It's option C only. Okay. Metabolism cannot yield free glucose. That is true. Muscle lacks glucosic phosphatase. So this is true. Third, oxidation of pyruvate to carbon dioxide occurs exclusively in the mitochondria. Can occur under aerobic conditions. Involves intermediates that are also involved in amino acid catabolism. Is regulated by concentration. Next. This is the 13 is the next question.
Mm, okay.
Hmm.
The oxidation of pyruvate to carbon dioxide. The oxidation of pyruvate to carbon dioxide, okay, occurs exclusively in mitochondria. Occurs under anaerobic condition. Involves intermediate that are also involved in amino acid catabolism. Is regulated by the concentration of acetyl CoA in the cell. Is impaired by thiamine deficiency states. Oxidation of pyruvate occurs exclusively in mitochondria. It occurs in because can occur in under anaerobic condition. Okay. Into involves intermediate that are also involved in amino acid metabolism. Also involved in mm, impedance, thiamine deficiency. TPP is option fifth is correct. Because exclusively in climate condition. Intermediate that time catabolism. For option B,
So you mean to say second one is false, can occur under anaerobic condition, the oxidation of pyruvate to carbon dioxide is option B is correct. Occurs exclusively in mitochondria. That is true. Pyruvate dehydrogenase complex is located in the mitochondrial matrix. Cytosolic pyruvate must be transported into the mitochondria for oxidation. Can occur under anaerobic condition. That is false. Pyruvate oxidation requires oxygen indirectly via NAD plus regeneration through ETC. Under anaerobic conditions, pyruvate is diverted to lactate formation. Instead, involves intermediate that are also involved in amino acid catabolism. That is true. Okay, intermediates such as acetyl-CoA, NADH, and TCA cycle intermediates overlap with amino acid catabolic pathways like alanine, leucine, isoleucine metabolism is regulated by the concentration of acetyl-CoA. That is true, and is impaired by thiamine deficiency state. That is also true. Okay, next.
plasma albumin binds the following. Plasma albumin binds the following. It binds free fatty acids, triglycerides, estradiol, bilirubin, iron ions. It binds to estrogen. Option 3 is correct. So D is ruled out. Triglycerides is also wrong. Free fatty acid, triglyceride, bilirubin, iron ions. Iron ions it doesn't binds. Option fifth is false. D was ruled out. Option false. So A, B, C now. Bilirubin, free fatty acid. Free fatty acid binds to the B, C. Iron doesn't bounce. Estrogen it bounds. Free fatty acid will bound. Now B and C, Mr. Phones are triglycerides and bilirubin. Triglyceride bilirubin it binds the 9. Option C, sir.
Options B.
Yes.
Free fatty acids, yes. Albumin is the main carrier for non-esterified fatty acids in the plasma. TGs is false. TGs are transported in plasma within lipoproteins, that is chylomicrons VLDL, not bound to albumin. Remember, FFA, yes. Free fatty acids, but triglycerides, no. Okay, estradiol, yes. Okay, bilirubin, yes. And ions are not. Okay, next.
Methionine is an essential amino acid. Is a sulfur-containing amino acid. Can be converted to cysteine by fetal liver. Can be converted to cysteine by fetal
Cannot be
liver.
converted. Cannot be
Cannot,
converted.
cannot be converted to cysteine by fetal liver. Cannot cross the placenta. Is reabsorbed in the proximal convoluted tubule in the kidneys. Option 2 and 5th is correct too. 2 and 5th. So option A, B, C cannot cross the placenta. Insulin is an essential. Insulin is an essential. First one is correct. Second one is correct. Fifth one is correct. And to fifth
So
is correct.
everything is on fourth only. Cannot cross the placenta or can. That is the deciding here. Okay.
Converted to system.
Let us go. The option B is correct. Okay. So is essential amino acid? Yes. Is a sulfur containing? Yes. Cannot be converted to cysteine by the fetal liver? True. The fetal liver has limited activity of transsulfuration pathway enzymes. So conversion of methionine to cysteine is insufficient. Hence cysteine becomes conditionally essential in fetal life. Okay. Then cannot cross the placenta, false. Methionine is actively transported across the placenta to support fetal protein synthesis. Is reabsorbed in the proximal convoluted tubule in the kidneys, that is true. Next.
Yes, sir. Tetrahydrofolic acid is involved in purine synthesis. THF is involved in purine synthesis. Okay, is a precursor of folic acid. Is a precursor of is a coenzyme in amino acid synthesis. Catalyzes the conversion of glucose to glucose 6 phosphate. Activity is inhibited by methotrexate. Option fifth is true. Is a precursor of folic acid. It is the folic acid. Option 2 is true. Fifth is true. Utilizes the conversion of glucose. Option fourth is wrong. Fourth is wrong. Option it is option is involved in purine synthesis. Yes, it is involved. Option A is precursor of folic acid. The coenzyme acid synthesis. Okay. Sir, option D.
D or B?
B.
It is B. Is involved in purine synthesis, that is true. THF donates 1 carbon units required for purine ring formation, C2 and C8 positions. Is a precursor of folic acid, that is false. THF is the active reduced form of folic acid and not its precursor. Okay. Folic acid is converted to THF in the body. Is a coenzyme in amino acid synthesis, that is true. THF participates in amino acid metabolism, especially serine, glycine conversion and homocysteine methionine pathway. Catalyzes the conversion of glucose to glucose 6 phosphate, that is false. This reaction is catalyzed by the hexokinase or glucokinase, not THF. Activity is inhibited by methotrexate, that is true. Okay. Next.
Deficiency of the following substances and diseases are correctly matched. Thiamine, pellagra, cyanocobalamin, microcytic anemia, okay, niacin, beriberi, folate sprue, ascorbic acid, night blindness. Actually, all are wrong. Yes. Option C, sir.
Option C. Thiamine, pellagra. See, this raid I have marked because malabsorption generally sprue is associated with malabsorption, not the
Hmm.
folic. It is associated, sprue definitely is associated with folic acid, but not exclusively for folic acid because it is a malabsorption. So folic acid is one thing, but not the exclusive, I mean to say, association. Okay. So
Hmm.
C is correct answer. Thiamine pellagra, that is false. Thiamine deficiency leads to beriberi and Wernicke's encephalopathy, Wernicke's Korsakoff's syndrome. Pellagra is due to niacin deficiency, B3. Then cyanocobalamin microcytic anemia, that is false. Vitamin B12 deficiency causes megaloblastic anemia. Okay. Niacin beriberi, that is false. Niacin deficiency will lead to pellagra, dermatitis, diarrhea, dementia. Folate sprue. Sprue, especially tropical sprue, causes malabsorption leading to folate deficiency, but the disease deficiency matching is not direct. Standard pairing in sprue is not a specific folate deficiency disease. Okay, classic folate deficiency causes megaloblastic anemia. Okay, ascorbic acid night blindness is false. Vitamin C deficiency causes scurvy. Night blindness is due to vitamin A deficiency. Okay, folate in sprue, yes, sprue commonly leads to folate deficiency due to malabsorption, but this is a consequence, not a specific classical disease deficiency match. Okay, next.
Biochemical abnormalities associated with diabetes mellitus include increased breakdown of protein, decreased plasma levels of free fatty acid, increased serum cholesterol concentration, decreased glycosylation of hemoglobin, or decrease in plasma concentration of LDL. Fifth is wrong. Fourth is also wrong. Third is true. Decreased plasma levels of free fatty acids.
So these two wrong and third is true. So we have B in this sequence. We have B and E only.
Hmm.
Now here only first two. Then you have to decide.
Decreased plasma levels of free fatty acids. No, it is increase. Option B, sir.
Likely to be B only. Yes. Increased breakdown of protein, that is true. Insulin deficiency causes catabolic state leading to increased proteolysis and amino acid release for gluconeogenesis. Decreased plasma levels of free fatty acid, that is false. In diabetes mellitus, there is increased lipolysis leading to raised free fatty acids, not decrease. Increased serum cholesterol concentration, that is true. Diabetes is associated with dyslipidemia, including increased cholesterol and TG due to altered lipid metabolism. Decreased glycosylation of hemoglobin, that is false. HbA1c represents increased non-enzymatic glycosylation of hemoglobin, not decrease. Decrease in plasma LDL concentration, that is false. Diabetes typically causes increased LDL, often small dense LDL particles, not decreased levels. Next.
Yes, sir. Concerning folic acid is a water-soluble vitamin. Conversion of DHF to THF is inhibited by methotrexate. Yes. It is a water-soluble vitamin A, D. Yes. Now, red cell folate concentration can be reduced by phenytoin. THF is a carrier of one carbon unit. It is involved in synthesis of purines. Option A is correct. B is correct. A, B correct. 2 A, B, D. Red cell folate concentration can be reduced by phenytoin. Yes, option third is also correct. So A and D. THF is the carrier of one carbon unit. THF is a carrier of one carbon unit. It is involved in the synthesis of folic acid purines. Actually, it is right. Fifth one is also correct. THF is a carrier of one carbon unit. Correct. Sir, option A.
All are correct. Okay, it is a water soluble vitamin. Conversion of uh, dihydrofolate to tetrahydrofolate is inhibited by methotrexate, that is true. Then red cell folate concentration can be reduced by phenytoin, that is also true, because long-term phenytoin therapy interferes with folate absorption and metabolism, leading to reduced red cell folate levels. Tetrahydrofolic acid is a carrier of one carbon unit, that is true. THF carries methyl, methylene, methinyl, and formyl and formino groups in numerous metabolic reactions. It is involved in synthesis of purines, that is true. Folate coenzymes donate one carbon units required for de novo purine synthesis and DNA replication. Next.
Yes, sir. Cholecalciferol, vitamin D, promotes the absorption of calcium from the gut. Is 25 hydroxylated in the liver. Is synthesized in the skin. Is 1 hydroxylated in the kidney. Is most active in the 125 dihydroxyl form. So all are true. So option A.
Yes, all are true. Promotes the absorption of calcium from the gut. Yes. Then in is 25 hydroxylated in the liver. That is also true. Then synthesized in the skin. UVB radiation converts 7-dehydrocholesterol in the skin to cholecalciferol. First hydroxylation in the kidney. Yes. Okay. Is most active in 125 dihydroxyl form. Yes. Okay. So all vitamins, please read. Okay. Next.
Yes, sir. Chemicals are solubilized in the liver by glucuronide formation, transamination, bile salt conjugation, acetylation, carboxylation. Except third, all are true, sir. So, third, not bile. Okay.
Option C. Glucuronide formation. Yes. Glucuronidation is a major phase 2 hepatic detoxification reaction. Conjugation with glucuronic acid increases water solubility and promotes urinary or biliary excretion. Then transamination is false. Transamination is involved in amino acid metabolism, not in hepatic solubilization or detoxification of xenobiotics. Bile salt conjugation is also false. Okay. Bile salts themselves are conjugated with glycine or taurine, but conjugation with bile salts is not a general mechanism for solubilizing drugs or chemicals. Okay, so here we are talking about external chemicals. So transamination is not there, bile salt conjugation is not there. Yes, acetylation, any drug can get metabolized by acetylation. Okay, and this first, okay, glucuronide formation. Okay, then carboxylation is false. Carboxylation is involved in specific metabolic pathways like clotting factor synthesis, pyruvate carboxylation, and is not a major hepatic solubilization mechanism. So the major hepatic phase 2 conjugation reactions are glucuronidation, acetylation, sulfation, methylation, glutathione conjugation, and glycine conjugation. Okay. So transamination is amino acid metabolism, not detoxification. Carboxylation is metabolic activation pathway, not solubilization. Next.
Messenger ribonucleic acid mRNA is a double-stranded polymer. Is transcribed from DNA in the nucleus. Is not present in reticulocytes. Contains thiamine. Is not present in oocytes. Option 4 is wrong. Is
Correct.
transcribed from option 2 is correct. Correct. Fourth is wrong. Option A, B, D, A, B, E. It's not present in thiamine is wrong. Okay.
mRNA is a double stranded or single stranded?
mRNA is a single-stranded polymer.
Yeah.
So you can
So
see.
we have only three options here now. B, C, D only.
So it
Okay.
is present. It is not present in oocytes. No, it is present. So contains thiamine. No, it is not contained. So from B and C is not
From
present
B
in
and C. Then
reticulocytes. Related are basis to option C, option B, option B.
answer is option B. Okay. So it is double stranded is a false. Transcribed from DNA in the nucleus, yes. Is not present in the reticulocytes, false. Contains thymine, that is also false. Is not present in oocytes, that is also false. Oocyte contain abundant maternal mRNA, which is essential for early embryonic development before activation of embryonic genome. Okay. So common RNA from examination point of view, DNA contains deoxyribose and thiamine. Okay, RNA contains ribose and uracil. Then reticulocytes lack a nucleus but retain mRNA and ribosomes. Oocytes are rich in stored maternal mRNA required during the first stage of embryogenesis. Next.
Yes, sir. Ketone bodies can be utilized by the fetal brain, includes acetoacetate, are water soluble, are synthesized in skeletal muscle, can be utilized during starvation. Option B is true. Option 2 is true. Option D is rule out. Are synthesized in skeletal muscle. Synthesized in skeletal muscle. It is synthesized in liver. Option 4 is wrong. B, C, D. Can be utilized by the fetal brain. Yes, it can be utilized by the brain. Two. Ketone bodies. So option E.
Option B is correct. can be utilized by the fetal brain. That is true. The fetal brain can utilize ketone bodies as an energy source for lipid synthesis during development, include acetate. That is also true. Then are water soluble. That is true. Are synthesized in skeletal muscle. That is false. Ketone bodies are synthesized primarily in the liver mitochondria from acetyl CoA derived from fatty acid oxidation. Skeletal muscle utilizes ketone bodies but does not produce them, and can be utilized during starvation. Yes, during prolonged fasting starvation, ketone bodies become an important fuel for the brain, heart, and skeletal muscle, reducing glucose requirements. Ketone bodies are produced in the liver but not utilized by the liver. Okay, they are synthesized in the liver but not utilized by the liver because hepatocytes lack the enzyme thiophorates, succinyl CoA, and acetoacetate CoA transferase, which are required for ketone body oxidation. So it is produced by the liver, used by muscle and brain during starvation, and not synthesized by the muscle. Okay, there are some older physiology books which mention that ketone body forming enzymes may be present in the extrahepatic tissues including muscle. Okay, so trace ketogenesis can occur experimentally, so muscle may synthesize theoretically. Okay, then liver is the site of synthesis for ketone bodies. Muscle, brain, heart will utilize the ketone bodies. Therefore the statement are synthesized in the skeletal muscle, what we have said, it is a false. Okay, next.
Noradrenaline is a noradrenaline is a derivative of arginine. Is a precursor of adrenaline. Adrenaline acts predominantly on alpha receptors. Is catabolized to VMA. Is metabolized by COMT. Sir, option A one is wrong. Adrenaline is a precursor of adrenaline forms noradrenaline. Is a precursor of adrenaline. Option B is right. Okay. Is acts predominantly, or if you know it is wrong, is catabolized to VMA. Metabolized by COMT. Fourth and fifth is right. Fourth, fifth is right. Third is wrong. That's because of adrenaline. Sir, option C.
Option B is correct. Is a derivative of arginine. That is false. Noradrenaline is synthesized from tyrosine. So tyrosine to dopa, dopa to dopamine, dopamine to noradrenaline, noradrenaline to adrenaline. Okay. Arginine is the precursor of nitric acid, not noradrenaline. Is a precursor of adrenaline. Yes. In the adrenal medulla, phenylphthaloamine and methyl transferase PNMT converts norad to adrenaline, acts predominantly on alpha receptors. That is true. He is catabolized by to vinyl
Yes.
mandelic acid. Yes, VMA. Then he is metabolized by catechol or methyl transferase. That is also true. Okay. So catecholamine synthesis pathway: tyrosine to dopa, dopa to dopamine, dopamine to norad, norad to adrenaline. Major enzymes are tyrosine hydroxylase, dopa decarboxylase, dopamine beta hydroxylase, and PNMT. Major metabolites are adrenaline, noradrenaline will be metabolized to VMA, and dopamine to HVA, that is homovanillic acid. Next.
Nitrogen balance is positive during pregnancy, positive during prolonged immobilization, negative during protein starvation, negative in the untreated diabetes, positive during recovery from debilitating illness. Nitrogen balance is positive. First one is true. Is during prolonged immobilization is also true. Nitrogen balance negative during protein starvation. Positive during recovery. Option B, sir.
Option B, nitrogen balance, that is the nitrogen intake minus nitrogen loss. Positive nitrogen balance, intake exceeds loss. Okay, occurs during growth, pregnancy, recovery from illness, and anabolic states. Negative nitrogen balance, that is the loss exceeds intake, occurs during starvation, uncontrolled diabetes, trauma, burns, and catabolic status. Positive during pregnancy, yes. Positive during prolonged immobilization, false. Immobilization causes muscle wasting and increased protein breakdown, producing negative nitrogen balance. Okay. Negative during protein starvation, true. Okay. Negative in untreated diabetic, true. Then positive during recovery from debilitating illness, true. Positive nitrogen balance will be seen in pregnancy, childhood growth, recovery from illness, and anabolic hormone excess. Negative nitrogen balance, starvation, protein deficiency, uncontrolled diabetes mellitus, trauma, burns, sepsis, and prolonged immobilization. Next. Yes, next. Yes, doctor, can you see it?
Hello, hello. Sorry, I
Yes,
forgot to
can you see
unmute.
the
Yes, sir.
next?
Yes, yes, sir. The following are amino acids found in human protein: serine, methionine, tyramine, histamine, arginine. Serine,
Human
methionine,
proteins.
human protein, serine is there, methionine is there, tyramine is not there, histamine and arginine. Option D, sir.
Option
D.
B.
Histamine is
B
also
means
there.
what is not there? Uh,
Hmm.
Third and fourth,
Meth.
tyramine and histamine. Okay,
Hmm.
so serine, yes, serine is a one of the 20 standard amino acids incorporated in human proteins. Methionine, yes. Tyramine is false. Tyramine is not an amino acid found in the proteins. It is a biogenic amine formed by decarboxylation of tyrosine. Then histamine is also false. Histamine is not a protein amino acid. It is a biogenic amine produced by decarboxylation of histidine. Okay, then arginine, yes, arginine is a standard amino acid found in the human proteins. Next.
Yes, sir.
Co-
Concerning the genetic control of protein synthesis, mature mRNA contains introns. A codon has three base sequence. Each amino acid has a single codon. Transfer tRNA has anticodon, tRNA anticodon recognition sites. Okay, each tRNA carries a specific amino acid. The fifth one is correct. Carries a specific amino acid. Each tier transfer has anticodon. Fourth one is also correct. Each amino acid has a single codon. Codon has a three. Second one is correct. Mature mRNA contains introns. No. Mm. Answer option B.
Option B is correct. Mature mRNA contains introns. That is false. Introns are removed during RNA splicing. Mature mRNA contains only exons. Okay. A codon has three base sequences. That is true. Each amino acid has single codon. That is false. A genetic code is degenerate. Most amino acids are encoded by more than one codon. Only methionine AUG and tryptophan UGG have a single codon. Transfer RNA has anticodon recognition site, true. Okay, each tRNA carries a specific amino acid, true. So from examination point of view, codon equal to triplet on mRNA. Anticodon, that is the complementary triplet on tRNA. Mature mRNA has got only exon. Introns are removed. One tRNA will code for one specific amino acid, but several tRNA may carry the same amino acid because of codon degeneracy. Then most amino acids have multiple codons, degenerated genetic code. Next.
Concerning nitrogen metabolism. Ingestion of complex proteins is essential for the maintenance of nitrogen balance. There are 8 essential amino acids. The minimum daily requirement of each essential amino acid is 0.3 to 1 gram. Amino acid nitrogen is largely excreted as urea. A protein poor but energy sufficient diet will cause a decreased excretion of creatinine. Option A is correct. essential, semi, it also includes semi-essential. Hmm. Hmm. Sir, option D.
Option
D.
B is
D.
correct. Okay. Next.
The following are disaccharides: glucose, maltose, sucrose, fructose, lactose. Sucrose is not. Sucrose and maltose. 1 and 4 is not there. Maltose, sucrose, and lactose is the option B, sir.
What? B
Glucose is
uh,
monosaccharide.
and
Lactose and fructose.
fructose. Fructose
Fructose
is
is
a,
also monosaccharide. So 2,
yeah,
3, 5 are
yeah.
positive.
Option B is correct. Okay. Option B is correct. So glucose is false. Glucose is a monosaccharide. Maltose is true. Maltose is disaccharide. Two glucose molecules will form maltose. 1,4 glycosylated bond. Sucrose, yes. Glucose plus fructose. Fructose is a monosaccharide, and lactose is disaccharide composed of galactose and glucose. Glucose, fructose, and galactose. These are monosaccharides. Maltose is 2 glucose. Sucrose is glucose plus fructose, and lactose is galactose plus glucose. Okay. So glucose and fructose are monosaccharides, whereas maltose, sucrose, and lactose are disaccharides. Next.
The following are actively transported across the placenta from the maternal to the fetal circulation: histidine, alcohol, ascorbic acid, iron, intrinsic factor. 3 and 4 is true, and 4 have A, B, C. Alcohol, so option B.
Option B is correct. Histidine is, yes. Histidine is an amino acid, and like other amino acid, is transported across the placenta by active transport mechanism to support fetal growth and protein synthesis. Alcohol is false. Ethanol crosses the placenta by simple diffusion, not by active transport. Fetal blood alcohol concentrations can rapidly approach maternal levels. Okay, because it is a passive transport. Okay. Then ascorbic acid, yes. Vitamin C is actively transported. Then iron, yes. Intrinsic factor is false. Intrinsic factor is a gastric glycoprotein involved in the maternal vitamin B12 absorption. It is not transported across the placenta. So actively transported are amino acids, iron, calcium, ascorbic acid, folate. Then cross mainly by diffusion, oxygen, carbon dioxide, alcohol, and many drugs. Okay. And do not cross intact molecules, intrinsic factor, and most maternal proteins. Next.
Urea is formed mainly in the kidney, contains 2 amine groups, is formed by transamination from ketoglutarate, is an end product of nitrogen metabolism in the fetus. Excretion is unrelated to protein intake, formed mainly in the kidney. Option 3 is correct. Transfer ketoglutarate groups, glutamate plus ammonia.
Yes.
And option
Option A is correct. Okay. So in traditional physiology question banks, fetal nitrogen waste products are considered to be transferred to the maternal circulation for elimination. Therefore, urea is not regarded as a fetus own end product of nitrogen metabolism, and the statement is marked false, despite fetal urea production occurring biochemically. Okay. Urea cycle facts: site is liver, detoxification of ammonia. Nitrogen sources are one nitrogen from free ammonia and one nitrogen from aspartate. Formula is CONH2 2 times. Urea is formed in the liver, not in the kidney. Urea excretion increases with protein intake, and fetal urea crosses the placenta and is excreted by the mother. Next.
Mother. Vitamin E can be synthesized in the skin, is derived from the beta carotene, is toxic to tissues in high doses, is a teratogen, is required for rhodopsin synthesis. Option 3 and 4 is correct. Option is required for rhodopsin synthesis is also true. 3, 4, 5th is correct. It can be synthesized in the skin. Retinol, no. Derived from beta carotene. Sir, option B.
Yes, option B is correct. Can be synthesized in the skin is false. Unlike vitamin D, vitamin E is not synthesized in the skin, is derived from beta carotene. True. Is toxic to tissues at high doses. True. Is teratogen. True. Is required for rhodopsin synthesis. True. Okay. Vitamin A functions: vision, maintenance of epithelial tissues, growth and development, immune function. Deficiency will lead to night blindness, xerophthalmia, keratomalacia. Excess will lead to headache, hepatotoxicity, bone pain, teratogenicity. Next.
The following are essential amino acids: valine, thiamine, cysteine, glycine, and lysine. Thiamine is there. I have read this, but I can't remember it.
Essential amino acids. In fact, I think I have given some mnemonic also for this.
I've read that. Yeah.
Okay, let's see. B is the correct answer. So, lysine and
Valine.
valine. Okay, valine is true. Thiamine is false. Then cysteamine false. Then glycine is false, lysine is true. Using traditional physiology classification, essential amino acids: valine, leucine, isoleucine, lysine, methionine, phenylalanine, threonine, and tryptophan. Okay, some older textbooks additionally include histidine and arginine. Okay, particularly during growth. So thiamine is a B1 vitamin, not amino acid. Glycine is non-essential amino acid.
Yes, sir. Plasma LDL are less dense than chylomicrons. Predominantly contains non-esterified fatty acids. Are the main source of cholesterol for steroid synthesis. Contain more protein by dry weight than HDL. Attaches to specific cell membrane receptors. Oh, both is wrong. We'll get this. Less dense than chylomicron. Oh, shit. Option B or C, Mrs. Anything.
Option
B
D.
or C.
Yeah. Okay. So are less dense than chylomicron. That is false. Chylomicrons are the least dense lipoproteins because they contain. This question: are less dense than chylomicrons. That is false. Predominantly contain non-esterified fatty acid. That is also false. Are the main source of cholesterol for steroid. That is true. Okay. Then contain more protein by dry weight than HDL. That is false. And attached to specific cell membrane receptors. That is true. Okay. So lipoprotein order is chylomicron, then VLDL, then IDL, then LDL, and then highest will be the HDL, the smallest. Okay. So HDL will be the actually, whether this is correct or wrong. Chylomicrons are the smallest one, no? Then chylomicron, VLDL, they are TGs. LDL, cholesterol. Okay, and HDL is a will contain main protein. Okay. Free fatty acids are transported by albumin, not LDL. HDL contains more protein than LDL. LDL delivers cholesterol to steroid producing tissues. Next.
Yes, sir. Enzyme activity can be modified by concentration of the substrate, concentration of the product, pH, temperature, concentration of the coenzyme. We all are correct, sir. Option A.
Yes. So concentration of substrate, concentration of product, pH, then temperature and concentration of coenzymes. Okay, all are so major factors affecting the enzyme activity. These are the things what we have seen. So common example, enzyme activity is not fixed. It is influenced by both chemical environment, pH, temperature, and substrate or cofactor availability. Next.
Yes, sir. Fatty acids reaching the liver from the fat stores may be converted to glucose, conjugated with sulfate, metabolized in TCA cycle, incorporated in endogenous triglycerides, converted into ketones. Okay, option fifth is correct. Into endogenous triglycerides. It is also correct. Metabolized in the TCA cycle. Fatty acids conjugated with sulfate. No, converted to glucose. It can be converted to glucose. Fatty acid converted to glucose. Sir, option B or D? B.
B. Converted to glucose, that is false. Fatty acids cannot be converted into glucose in humans because acetyl CoA produced by beta oxidation cannot be converted back to pyruvate or yield a net synthesis of glucose. Okay. Then conjugated with sulfate, false. Metabolized in the TCA cycle, true. Incorporated into endogenous TG, true. Converted into ketones, true. Okay. So major hepatic fates of fatty acid, beta oxidation, that is converting to acetyl CoA. Acetyl CoA, then TCA cycle. Then acetyl CoA can get converted to ketone bodies, and re-esterification will lead to the TGs and VLDL. Okay, fatty acids cannot be converted to glucose in humans. Glycerol, not fatty acids, contribute significantly to gluconeogenesis. Next.
Yes, sir. Cholesterol is synthesized in the liver. A C19 compound synthesized from acetate, a major constituent of HDL, predominantly excreted unchanged in the urine. Option B is wrong. Option fourth is correct. Synthesized from the acetate, acetoacetate. True. A C19 compound synthesized in the liver. Option B, sir.
Option C.
C.
Okay, it is synthesized in the liver. That is true. C19 compound, it is false. Cholesterol contains 27 carbon atoms. Okay. Then synthesized from acetate. That is true. Cholesterol is synthesized from acetyl CoA via HMG CoA reductase pathway and a major constituent of high density lipoproteins. False. Predominantly excreted unchanged in the urine. That is also false. So cholesterol facts, it, how many carbon atoms? 27. Precursor acetyl CoA. Rate limiting enzyme HMG CoA reductase. Then precursors of steroid hormones, bile acids, vitamin D. So from examination point of view, cholesterol is not a C19 steroid. Cholesterol is not excreted in the urine. HDL is characterized by high protein content, not predominantly by cholesterol. Next.
The level of serum uric acid characteristically falls with starvation, is higher in men than women, rises on taking corticosteroid therapy, falls on treatment with 5 gram of aspirin daily, increases in acute leukemia. Yes, fourth one is wrong. Falls on treatment serum aspirin. Hyperuricemia के treatment. Aspirin causes hyperuricemia. Option C, sir. Option B.
Okay, false with starvation, that is false. Okay, starvation increases the ketone body production. Ketone compete with urate for renal excretion, leading to hyperuricemia, especially during early starvation. Is higher in men than women, that is true. Rises on taking corticosteroid therapy, false. Corticosteroid generally increase renal urate excretion and are not characteristically associated with hyperuricemia. False on treatment with 5 grams of aspirin daily, that is true. because high dose of salicylate, that is about 5 gram per day, are uricosuric and will lower the serum uric acid. Okay, low dose aspirin has opposite effect and may raise the serum uric acid. Okay, increases in acute leukemia, that is true. Causes of raised uric acid: starvation, fasting, acute leukemia, tumor lysis, thiazide diuretics, renal failure, and gout. Effect of aspirin, low dose will reduce the urate excretion, will increase the uric acid. High dose around 5 gram will be uricosuric and decrease uric acid. Next.
L-glucose is a pentose and ketose, and aldose, a ketose, a mirror image of D-glucose, identical in biological activity to D-glucose. 4 is correct. The pentose, aldose. So option D.
Option A is correct.
A.
Is it a pentose? No. Glucose contains 6 carbon
Sir,
atoms.
your voice is less.
Yeah.
Can you increase your volume?
Option A is option A. Sorry, pentose, that is a false. Glucose contains 6 carbon atoms. Is therefore hexose, not a pentose. Aldose, it is true. Glucose is an aldohexose. Okay, containing aldehyde group in the open chain form.
Okay.
A ketose,
Mhm.
false. Ketoses contain ketone group like fructose. Glucose is a aldose, not ketose. A mirror image of
Mhm.
D glucose, that is true. Okay. Then identical in biological activity to D glucose, that is false. Okay. Most biological enzymes and transport systems are stereo specific for D glucose. L glucose is not metabolized normally and therefore does not have identical biological activity. Okay. They may be mirror image, but the biological activity will be different. Glucose is aldohexose, galactose is a aldohexose, fructose is a ketohexose, ribose is a aldopentose, and deoxyribose is a aldopentose. Okay, so D and L glucose have identical chemical formulae and physical properties, but they do not have identical biological activity because enzymes and transport proteins are stereospecific. Okay, next.
Folic acid requires gastric intrinsic factor for its absorption. Daily requirement is about 50 mg.
40.
Is found in higher concentration in fetal blood than maternal blood. Deficiency leads to microcytic anemia. Is fat soluble. Um, Deficiency leads to macrocytic anemia. Is fat soluble. Acid, intrinsic acid. Fourth one is false. Found in higher concentration in fetal blood than maternal blood. And one is also false. First, second, and fourth is wrong. So option A.
Yes. Requires gastric intrinsic factor for its absorption. That false. Intrinsic factor is required for vitamin B12 absorption, not folic acid. Folate is absorbed mainly in the proximal small intestine, that is jejunum. Daily requirement is 40 milligram, that is false. Normal daily folate requirements are measured in micrograms, not milligrams. Adult doses are 200 to 400 microgram per day. Increasing during pregnancy is found in higher concentrations in fetal blood than maternal blood. That is true. Folate is actively transported across the placenta, resulting in fetal concentrations that are higher than the maternal. Deficiency leads to macrocytic anemia. That is false. Is fat soluble. That is also false. So it is a water soluble vitamin absorbed in the jejunum, does not require intrinsic factor, required for DNA synthesis and one carbon transfer reactions. Deficiency will lead to megaloblastic anemia and increased requirements in pregnancy. Then intrinsic factor is for vitamin B12, not folate. Folate deficiency causes macrocytic and not microcytic. Folate requirements are in microgram, not milligram. Next.
Concerning glycolysis, it is the mobilization of stored glucose units. Most of the reaction occurs in the mitochondria. Two molecule of ATP are consumed per molecule of glucose. Yes. There is a net gain of two ATP molecules from the conversion of glucose molecule to two pyruvate molecules. The glucose to pyruvate pathway is present in all tissues. First one is true, second one is wrong. It occurs in cytoplasm. There is the molecules of ATP are consumed per, that is true. Of 2 ATP from the converted glucose molecule to 2 pyruvate molecules. Uh, false.
Glycolysis is
This
very
word.
important from the examination point of view.
Yes, sir. The glucose to pyruvate pathway is present in. First one is correct. The glucose is to pyruvate is present in all tissues. Yes. Present in. There is net gain of 2 ATP molecule from the converted. No, 7 ATPs are generated. No. See. Simple. Glucose molecule to pyruvate. To pyruvate molecule. So, sir, option A.
Option A is correct. Glycogenolysis is the breakdown of glycogen to glucose. Glycolysis occurs in the cell cytoplasm. Then the investment phase requires 2 ATP and the dividend phase 4 ATP. The net gain is of 2 ATPs. Okay. So that finishes the biochemistry records. Thank you.
Yes, sir.
So